your comments and suggestions. So once more time, we're going to go ahead and record the meeting. Please go ahead and mute your mic so we don't hear any background noise and we can hear our presenters loud and clear.
Igualmente, gracias, bye.
Awesome. Go ahead and keep your camera off, students. Please make sure you keep your camera off. And then if you have any questions or comments, obviously we encourage you to please use the chat box. You guys have been through these presentations before, so you know that we answer your questions and we'll respond to your comments throughout the presentation or towards the end. For students, please make sure you sign in using the link that's in the chat box. Some of you are probably getting extra credit, but we need to make sure your attendance is tracked. So please make sure you sign in. And for our CT teachers who are here, please be sure you look out for a message from Ripsame. You're required to go ahead and sign in right now at the top of the hour and also sign out at the bottom of the hour. Please make sure you also continue to mute your mics, please. Thank you. And with that being said, welcome health science and medical technology students and CT teachers. My name is Sarai Franco, and I'm here with my colleagues, Ripsame and Richard. You know us by now, and one more time, we're bringing you another event. But again, we're your work-based learning coordinator support team. So we welcome you to our second event from our COVID series that is brought to you by the Crest Institute of Science. And today's presentation will be on the immune system and its vaccines. So we will be discussing the immune system with a focus on adaptive immunity. We will also be providing a background of all the vaccines and details of novel vaccines against COVID and their efficacy. So with that being said, I'll let Dr. Webster go ahead and take it away and introduce Manji. Thank you.
Good afternoon, everybody. And thank you again to the team for inviting us here. Today, I would like to introduce you to my colleague, Manjula Gunawardana, and he is going to talk about adaptive immunity and vaccines. And to do him justice for an introduction, I would like him to perhaps tell us where he's from, how he got to Oak Crest, and what sort of education he's got that allows him to talk about it, adaptive immunity and vaccines. Okay, Manji, over to you.
All right.
They're a fun team.
Thank you, Paul. Thank you very much. And thank you for the LAUSD team, Richard, Sarahi, and rest of the team. It's my pleasure to meet all of you. As Paul mentioned, to give you a little background, I finished my high school in Sri Lanka. We were just talking before you joined. It's a small island in the Indian Ocean, okay? And then when we moved here, I was at Pasadena City College, which is a two-year community college in Los Angeles area, as you might already know. And then I transferred to UCLA. And in addition to my undergrad, I started doing research over there as well. And while there, I applied to medical school and got selected to UC Davis and deferred it for 1 year to join a small biotech company. And that was it. I stayed with the biotech company, but pursued my higher education with a master's in public health at UCLA, and then worked at biotech, which we merged with a mid-sized pharma company for about 10 years. And I was a group leader there doing enzyme development. So I was doing a lot of DNA, RNA manipulation, gene manipulation and synthesis. Then about 10 years ago, I joined Mark Baum and Oak Crest. I got to know Mark while I was with the pharma, with the industry. I used to bring in Mark's interns to my lab. And then since I joined Oak Crest, we started our micro and molecular biology lab. And it's an esteemed faculty to work with. And now I facilitate micro and molecular biology work and do biomolecular drug release at Oak Crest. And also with Paul's expertise and leadership, I help out on these outreach programs as well. So that's it. Hopefully, the bottom line is keep pursuing what you want to do, and you need to find a happy niche that you'll be content, and start working from there. I hope that's good enough. You can ask me questions later, and can I go for the presentation now? Is that okay?
Go right ahead.
All right. So I was volunteered to do 2 presentations. So I would initially discuss about the immune system, human immune system, specifically focusing on adaptive immunity and some of the vaccines. And later on the second presentation, I think it was the end of this month, would discuss a little bit on the antigen and antibodies that promote some of these vaccines, the science behind that as well. Just to give you an introduction on what is on the first slide, this is a computer graphic from one of our collaborators at Scripps Institute in San Diego. And the structure, the gray structure, is actually a HIV virus, and they are working on antibodies that bind to these viruses. So the colored molecules that you see are actual antibodies binding to specific sites on that virus. So I got this picture from one of our collaborators and thought I would share it. I don't think it's published yet, but that's why I don't give any credit yet, right? Okay, so to start with the immune system, you can approach the immune system to study the system as innate immunity and adaptive immunity. Innate immunity is what is on your, the physical, hold on one second, the physical, I'm trying to move some of these screens, one second. So we have physical barriers on our body that act as initial defense mechanism. They are like the skin, which is the largest organ in the human body. The skin covers us, it's wrapping around our body that keeps invaders from entering into the body. And then you have saliva in your mouth, and then the flushing mechanisms of urine and tears, and then you have stomach acid inside your digestive tract. If you visualize your digestive tract, it is actually an open tube, and what is within that tube is pretty much outside. So you have that acid chamber that can catch and destroy some of these microbes or invaders, pathogens, before it gets into our body. So overall, the physical barriers, the responsibility is to stop invaders or pathogens from entering the body. Then we have another set of defensive mechanisms based on our blood in the circulatory system. We have differentiated specialized cells. So you have, probably you understand them as immune cells. These are different types of immune cells, the phagocytes, the neutrophils, macrophages, basophils, eosinophils, and natural killer cells. So these are specialized cells that are in our circulatory system that move from place to place, and they directly interact with the pathogens or the invaders to the human body and get rid of them by killing them. And basically killing in the sense of digesting that cell, the pathogen, so they no longer get into our cells within the body. So the next slide is on the adaptive immunity, which we'll focus a little bit further. So adaptive immunity can be separated into 2 separate groups again. are T cell immunity and B cell immunity. You probably heard about these as white blood cells or immune cells. And T cell immunity happens when you have an antigen or that invader, the molecule that creates an immune response. So that is an antigen. And when you are exposed to an antigen, maybe it's a pollen, maybe it's a shellfish, maybe it's a virus, maybe it's a virus called COVID these days. When you get exposed to it, the body reacts in a certain cascade of reactions and create antibodies against that specific pathogen. And then it gets into your circulatory system and blocks the invader from doing further damage to the body. T cell immunity goes hand in hand with the B cells. Okay, these are again another white blood cells or a set of immune cells. You can categorize them as helper cells, cytotoxic cells, and regulatory cells. And they too work in a complex manner. That end result is death of the infected cells that have already the pathogen has gotten into, or damaged cells that the pathogen is acting upon. So to go further into the adaptive immunity, you remember how I talked about the macrophages. So the macrophages can engulf or get the pathogen or the microbe into the macrophage, and then it digests and breaks up the pathogen, and it leaves little pieces like signatures on the periphery or the outside of the macrophage. So the other cells that we discussed, the B cells and the T cells, can interact together and make our immune response more efficient. And the B lymphocytes are the white blood cells I mentioned about, and they identify those pieces left behind by the phagocytes. And their specialty or the special responsibility is to create antibodies. T lymphocytes, again, as I said, it's a set of white blood cells. So the helper cells interact with the infected cells and produce different chemical molecules that can communicate with the rest of your body, especially with your immune system. And cytotoxic cells, cytotoxic, it means it attacks the infected and it breaks up the infected cells. And that's the regulatory cells, who are like the monitor, or you all are in high school. I don't know, do you still have to get a hall pass to go around at school? So they are the ones who are making sure our immune system doesn't overreact, because having an active immune system is important, but having an overreactive immune system is not beneficial. So those are the regulatory cells that say, okay, slow down, don't make too many cytokines, otherwise you are going to have an overblown reaction. If you are familiar with allergies and sometimes certain secondary states of infections, you have heard about COVID recently. I'm assuming everybody's aware of it now. And the secondary nature of COVID infection is you have these cytokine, chemokine storms happening in your body that takes over the immune system, and your body is using up so much energy that it becomes detrimental if you have other side effects. So you could be a heart patient or a diabetes patient, that the secondary infections takes place, maybe a pneumonia takes place, because what the virus does is they kill some of these T cells, and the regulatory cells, there's no longer monitoring of it. So you go into these overblown reactions that actually is detrimental and fatal. Just to give you a visual, So let me, hold on one second. So if you can see the red marker, if you take a virus as your pathogen or the invader into the body, the phagocytes will take in that specific pathogen and break up the pathogen and leave these little signatures. Now, whenever you see a schematic like this, it's very easy to say, okay, there's an arrow here go from A to B, there's an arrow like this. These are biological reactions. You have to give recognition to the complexity of those biological reactions. This is not one reaction. These are thousands of reactions that are happening in your body. So for simplicity, we draw these schematics and put arrows and say, oh, it's going from B cell to antibodies. This is a lot of reactions that we try to capture. So saying that it's a complex reactions happening here, but it ends up some of these proteins or glycoproteins, some of these biological molecules are expressed on the cell membrane of the phagocytes, and then it recruits T helper cells. So the T helper cells then send out messages to the B cells and other cytotoxic T cells. Now it generates a cascade of reactions that specific for those cell lines. Just to emphasize on it, because we will discuss a little bit more about antigens and antibodies on the next presentation. So the first time you get exposed to an antigen or a pathogen in this case, you are getting engulfed or taken in by the phagocytes, and they're presenting those antigens on the cellular membranes. And as the previous graphic showed you, they enlist B cells and the cytotoxic cells. And the T cells differentiate into memory cells. These are specialized T cells that in the long immunity, this is what causes longer immunity when you have an exposure. So it's beneficial because the next time you get exposed by the same pathogen, in addition to this stage, the first primary stage, your secondary compartments are ready to go. You have been exposed, you have the memory cells that quickly break in to the B cells cascade as well as the T cell cascade. So this method or the simplicity of this schematic will help you when we discuss about how some of the vaccines work as well. To give you an idea time-wise, you are going to get an infection, in this case, a viral infection. So within a couple of days or a week or so, you have a very high bioavailability of that specific pathogen or the antigen in your body. So it takes a few days for some of your immune responses to take place. That's why you hear what is the lag time for COVID. They would say like, okay, 3 to 5 days. What is the lag time for influenza virus? Maybe 1 to 3 days. So the virus or the pathogen has an advantage. Unless you have been exposed before, your body takes a couple of days to generate this cascade of immune responses. And so initially, you can see there's a spike in the specific antibodies. Remember, the antibodies are made by the B cells. And then gradually it falls off, or the concentration of bioavailability of those antibodies decrease, because now your body has started an immune response against this virus, and you can see how the virus numbers are going down, and it's no longer necessary, or it's not getting triggered by these macrophages and other immune cells to create more and more antibodies. So after a while, within months, on some diseases, maybe during years, the antibody levels go down. And then you have a specific T cell forming as well. So that there is a increase in specific T cells for that antibody, but after that, these T cells differentiate into the memory cell, and then they retain a lower number, gets retained in your body. And you can also see some of the B cell, memory B cells also, they have a little spike, but within months, they worn off because there's no necessity or there's no stimuli for those specific cells in your body, because this is assuming your virus has been attacked and it's no longer infecting other cells. Now, these charts and these graphs change depending whether you have an acute infection or you have a more chronic infection, because then if you have a chronic infection, you will not have this bottoming down, the tail down of the virus. You will have more of something like this, right? Tailing off. So that means your body is constantly working on attacking this infection. If you have heard about HIV or AIDS patients, they are immunocompromised because your body's immune system is, for the terms like working overtime to attack those cells that are hiding in our human cells. So the body has to use up a lot of energy and keep on making this immune cascade. And that's why at some point you become immunocompromised. So you can have a chronic infection of HIV, and now your body is immunocompromised and you get a pneumonia attack, and that is something like this, but it's so acute infection. That is what becomes fatal. So the adaptive immunity, you can look at it another layer. So it's things are like, if you take an onion, the more you peel, you see different pictures of it, or you can cut an apple different ways, horizontal or vertical, you see a different picture. So this is another way of looking at adaptive immunity. You can look at as a natural immunity and as artificial immunity. Natural immunity, one of the aspects is passive. This is where the mother and a newborn or the infant, this is where the breast milk or the placental fluid, the transmission, mother's immunity get transmitted in to the fetus or the infant, and some of the antibodies get passed through as well. So the natural active immunity is because every day we get exposed with microbes, we get exposed to different pathogens. Maybe you are eating a certain chemical, and little doses, they actually create an immunity in you. So we are not, as long as somebody's not in a bubble, every day you touch a surface, you touch your skin, you don't know, you touch your eyes. As we said, like, remember how the passive immunity is supposed to be. You ingest something, maybe you're having a salad, it has some E. coli in there. That doesn't mean don't eat salads, do eat your vegetables, right? But you constantly get exposed to these pathogens and antigens, but your body has a way of dealing with it and also working on making it an advantage. So active immunity is where you are getting an immunity based on actual infection, actual exposure of that specific pathogen. So for artificial, you sometimes heard about people getting transfusions. You can get plasma transfusion. So if you have a plasma transfusion from a person who already had a certain disease, because what we are living in right now, maybe it's a COVID patient. A person who survived COVID would have generated those antibodies. Remember the chart where after a while you have a spike of antibodies. Now that person's plasma is when you take blood and centrifuge down all the cells out, and the proteinaceous part, we call it the supernatant, because the cells are heavier, they get into at the bottom when you centrifuge a sample. Centrifuging is spinning a sample at very high speeds. So all the heavy material is at the bottom, you call it a pellet, and the less denser or less heavy material is called the supernatant, and the proteinaceous part is at the top. It's like a red blood. You spin it down, you'll have a blood pellet and a straw color liquid on top, which is your plasma. it contains the antibody. So if you have a transfusion of plasma, so that second person who's getting the recipient of the plasma is actually getting these antibodies against that specific pathogen. So let's say they get, either they were already infected or now they're going to get infected. Your body immune system is getting a boost because of the antibodies you're getting passively. It's artificial, but passive because you are not making it active. So the active is the vaccines. Vaccines are what we'll discuss in the next few slides. And this is the way your body is now prompted to make those antibodies against a specific pathogen or immune antigen. So traditionally, you can separate vaccines into different categories. Some of them are called live attenuated vaccines. So these are weakened pathogens or germs. So you use one way of weakening is using irradiation. So you can expose the pathogen to UV light, and at a certain level, you can do tests to show that it's not going to make an acute reaction in your body. It's making a more subdued reaction in your body, because if it weakened a virus or a bacteria, and then you expose that to a person, and then the body starts making that immune response in a more controlled manner. So when you do weakened vaccines, they make a strong, long-lasting response. However, because of the strong response you are making, if you have a weakened immune system, remember what I told you about how some of the cascades go overboard or out of control. And if you have that, that's detrimental. So you don't want that. So if you have a weakened immune system, those people and people with organ transplants or long-term health problems, they are not the best candidates for this type of vaccine. And also they have to be kept in very cold conditions. So if you think outside of certain areas of US and outside of US, it's not the most convenient way of transporting them. An example of weakened attenuated is, most of you should have gotten all these vaccines, mumps, the MMR, the measles, mumps, rubella, and chickenpox vaccines. So a second group is called inactivated vaccines. These are actually completely killed or inactivated version of the germ. And it could be a virus, it could be a bacteria, mainly it's viruses that have been completely inactivated. So the immune response is not as strong as you attenuated the live virus, the vaccines, but to compensate for that, we have to give 2 doses so that people would get complete immunity. Example of that would be the hepatitis and polio vaccines. Speaking of 2 doses, this is more relevant for what you hear now, right? You hear about COVID vaccines and these inactivated vaccines. So you have an initial response. Remember that graph or the schematic I showed you, the initial exposure and the secondary exposure. So you have an initial exposure, the immune response or the antigens spike up, but after a while they go down with the time. And then you get a second booster. So now some of your cells are ready. Now it will generate a secondary reaction, and more of those memory cells will be created in your body. So that's why we need a booster for some of these inactivated, and as well as later on what we talk about these mRNA vaccines. Another subset of the vaccines are the subunits or recombinant of certain biomolecules of that specific germ. So if you have a hepatitis or a certain virus, you can make a specific protein or a sugar that is unique to that specific pathogen, and then expose the body in very low dosages, so your body starts making those antigens. So they also make a strong reaction, and they can be used pretty much with everyone in the community, okay? And they need a booster set as well. There are certain COVID vaccines in this category, the protein category, Novavax, which is not, it's still in phase 3. It's not approved in Europe or in US yet, but there is a Novavax protein-based vaccine being created right now for the pandemic. So, the traditional vaccines examples are hepatitis and pneumococcal diseases. The last traditional group that I would discuss is the toxic vaccines, which are specific toxic made by the pathogen. So it could be a bacteria or virus, but if they're a toxic molecule that is created by the pathogen that gives you an acute reaction, now we can make a vaccine based on a smaller dose of that very specific toxin and expose people with a controlled amount. So you build up a gradual reaction, and then these also need booster shots, diphtheria and tetanus, and we all, I'm assuming all of you are getting vaccinated appropriately, and these give us the opportunity to combine different toxins, different subgroups, so you don't have to get four different injections or jabs on your shoulder. They can be controlled combined at the correct dosages, and you get 1 or 2 shots as the booster, and that's the end of it for many, many years. Now we'll transit into a little bit about COVID or SARS-CoV-2 virus, okay, because we'll focus a little bit on the COVID-based vaccines. Just to give you, I know there's a lot of information. Hopefully this is not overloading that information. Just to give you a refresher. So it's a coronavirus that has very specific protein signatures on the outside. Sorry, I went one slide ahead. This is the envelope of the virus, and within there you can see these spike proteins. These days everybody's talking about spike proteins, and this is to show you how these proteins interact with the host cells. This is the ACE2 receptor, and the other receptor that we scientists have figured out. This is how it's binding in order to get into the cell. Saying that, again, I'm showing you a graphic with, I call it pretty pictures, but with arrows and a very structured mechanism. These are very complex, complicated, thousands of reactions that are happening, but the benefit of understanding, even though it's superficial, understanding what is going on, this is the diagram I'm going to discuss from, okay? So it's not just a one step, oh, suddenly the virus is inside the cell, here goes the new generation. It's not as simple as that. So once the virus is taken in to the cytoplasm, so you have a cell membrane, and within that you have the cytoplasm, and within the cytoplasm, this viral envelope is exposed, and the nucleic acids, in this case for COVID or coronavirus, it's a RNA molecule that gets released onto the cytoplasm. And if you are familiar with where the RNA main metabolism takes place is in the ribosomes, that's our protein factory in the cytoplasm. So the RNA gets directly into the ribosome and starts making active proteins specifically for that virus. So it's an advantage if you think about a virus molecule that has RNA, it doesn't have to cross into the nucleus. Getting into the nucleus is another step, going through a nuclear membrane. So if you remember the dogma of biology, you have DNA, which has all the information about the structures, the proteins that needs to be made within a body. And then you have the RNA, which is like the translator, which is the messenger that brings that information in a manner the ribosomes can understand. So it brings DNA to RNA, converts into RNA, and RNA is brought through the nuclear membrane into the cytoplasm, where it reaches the ribosomes, where now it gets translated into proteins or the functional message. So if you think about a book, I don't speak French, so if a book is in French, I would see there's information, but I would not understand it. So I need a translator. So that intermediary step is the RNA, and that tells, the RNA tells me, make this or do this, and that is the functional part of that is the protein, if you want to visualize it like that. So once the new generation of virons are made in the body, remember the previous slides we discussed, you have these immune cells or phagocytes that get the cells broken, the virons broken in, and assemble and present these specific signatures on their cell membrane, so you can have this cascade of immune responses. So we touch base a little bit on that, and we'll probably discuss a little bit on the next presentation as well. So this is a general idea of how the viruses multiply, the particles multiply with using the host mechanism, and how some of the cascade mechanism takes place. So when you make vaccines, it takes time, it takes money, it takes a lot of resources, and sometimes it ends up not getting into the market. As I said, I come from a little bit of industry background. So to give you an aspect, you can have like 100 different projects that you are spending millions of dollars on each project. At that point, you don't know which project is going to make success, but it's not trivial to make a decision, okay, this project is not going to work, let's go ahead with this specific one, because nobody has that crystal ball to tell you, okay, this is the project that's going to cure such and such a disease. And if you think of it in an industrial, in a business way, which is important as well, this is the product that's going to make the company millions of dollars. Nobody knows that. Anyways, so after many years, so you see like the polio vaccine, it's a lot of resources. And if you take a look at it, it took about 60 years for the actual polio vaccine to come through and come into the society. And Ebola and some of these vaccines, SARS-1 and MERS, they, after 17, 20 years, 5 to 6 years, there's still no specific vaccine. Now, you have to keep in mind, it's a business aspect too, right? It's supply and demand. If there is no demand, there is no funding. That without no funding, the scientists cannot sustain that research. So it's important for people to understand, be aware of what is going on. If it's HIV, now very few people talk about HIV, but that is still happening. Every like 30 minutes we talk here, there's like 20 or 200, forgive me for the exact number, of people actually get infected from HIV. So that pandemic is still there, that epidemic is still there. On top of that, we have COVID. Now we have, everybody's talking about COVID. I think I checked a couple of days ago, even though now we are talking about, okay, the numbers are down, everybody should just chill. There were like 1,100 deaths in US 2 days ago. So these things are happening as students, as people who would take the message and become the next generation. I think all of you, irrespective of what you want to do at the end, specifically, you should be aware of things and get information from proper sources and make your own opinion and be involved. I know I'm going on a tangent, but I want to let you know, like, things are still going on, and this is how much effort people take, and sometimes there's nothing in there. So people have studied coronaviruses for many years. And then a year ago, we had a beginning of a pandemic. So there was more emphasis on that, and also a lot of resources going on to that. And because of that, we could have a vaccine by about December last year. We got the clinical trials went into March, and we had actual vaccine available for the first responders. I think December 18th was that significant day. We got a vaccine available, and now it's getting available for the rest of the population, including all the students who are already in school or going into school. So it took about a year and a half for these vaccines to be developed. So just to give you a rough idea about what a phase 3, so there are phase 1s, phase 2s, and phase 3s. Phase 1s are mainly based on safety of the vaccine in a very controlled, a smaller unit. Then you increase to look at the efficacy into a secondary group or a phase 2 group. And then phase 3 is where you expose that specific treatment to a large number, thousands of people. So if you take an example, the Mrna vaccine, which is now available in the US. So they had over 30,000 people who enlisted in this vaccine study, the phase 3 study. And it was pretty much all comers, means they did not specify, okay, this is the criteria to enroll in this study. So by opening up to everyone, you capture a vast variance of the population. Are they predisposed people? Do they have a secondary condition that can make a COVID infection more detrimental? Or are they completely healthy athletes or healthy people? And what is the age group? So they opened it up to all comers who enrolled on it, okay? And they had worked out the multiple dosages. Actually, there were 3 different dosages that they tested on phase 2, okay? And for the phase 3 trial, they decided based on the phase 2 data that it will be two doses, 21 to 28 days apart, okay? And then they had a control group that got a placebo, means they didn't get the active ingredients of the vaccine. They got a placebo effect on a placebo vaccine. That's how you compare how good or the efficacy of that vaccine. Okay, so the vaccine efficacy is in the 94, and for Pfizer, it was in the 95 range. That means compared to the placebo group that did not get the vaccine, there's a 94% chance that if you get exposed to COVID, the vaccine will prevent you from getting infected. And if you compare it to the rest of the community, 100% of the people who got the vaccine, they had very minor or like much mellower conditions when they got infected. So out of that 6% who got that infection comparatively, none of them were hospitalized. So if you had gotten the vaccine, let's say there's a 6% chance you get infected by the virus, even those 6% of the people, they do not get acute infections. It's much controlled. It's what they described is as flu. That was the condition that, and they were at home. They did not have to go into a hospital. They were not on ventilators. So the presentation of the symptoms were much less. So that's just to give you an idea of how a phase 3 trial. So now the changes are that you get an first jab or the first injection, and then after 14 days, you are ready for that booster. If you still have a visual of that graph, how the boosters, how the initial exposure, the antibodies go down, and then it go up on the second booster. you get a much higher response and lasts much longer. So the mRNA vaccines that we'll discuss, so they are RNA vaccines, and remember how the virus itself is an RNA, so it doesn't have to get into the nucleus, it is in the cytoplasm, and the mRNA message is for specific spike proteins that is exposed on the periphery of the virus. Remember, if you remember the structure of the virus, I think I have a, so if you have these spike proteins, so that is what the mRNA is coding for, the spike proteins, and the message is sent into the cytoplasm, and it doesn't get integrated into your genetic material in your nucleus, okay? And then once those spike proteins are exposed on the cell membrane of the immune cells, they create a T cell and a B cell cascade that generates immunity against the secondary exposure. Remember that graph where the secondary exposure, your body is ready to fight for the secondary infection. Now, even though it's the first infection your body was pre-exposed into, now your body is ready with all the memory cells that can quickly generate the immune response. So that's what prevents from the virus from expanding or populating within our body, so it doesn't become an acute infection where people have to get hospitalized. So the mRNA platform or the technique of making the viruses, they were used for Zika virus. If you remember a few years ago, especially in South America, there was a Zika outbreak, and then cancer, and now we use it for COVID-19. So the mRNA that is within the vaccine is coated with other material, or we call it excipients, that secures your package. So that way, when you get the injection, the mRNA doesn't get digested within your body before it enters the cell membrane. Again, this is another arrow. It's a complicated system, but the mRNA is incorporated into the cytoplasm, where now the excipients or the coating of the mRNA is removed and the mRNA is released. And then with another cascade of reactions, the mRNA gets into the ribosome of the cytoplasm, which is the protein factory of our cytoplasm, our cells. And then that mRNA is now translated into functional proteins. In this case, it's a spike protein, and now it is presented on these different cascades of immune cascades. So the cytoplasm is presented on the cell membrane, so those B cells and the T cells can begin more complex immune systems. Let me see. Okay, yeah, down to 10 minutes, I guess. So safety of it, as I said, it's not entirely new. It feels like it was rushed, but it took, because of the pandemic, it generated a lot of resources. People put in a lot of resources into getting, because of the demand, because of what was happening globally. We needed an answer, and when you put in a lot of effort, the outcome in this case was successful. So, and sometimes people discuss about the safety, how this is going to get into your cytoplasm, into a nucleus and make mutations. So this is a mechanism where the mRNA is not going into your nucleus. So there's no integration, and the mRNA innately has a certain type, because your body doesn't want to keep so many mRNA molecules. Remember, the mRNA is made by the host to make functional proteins in our body too. If you keep for a longer time, the body would not have resources. We'll be making just one protein, and that's all. Because of that, the mRNA gets recycled. So the half-life of the mRNA is in this vaccine, it's about 24 to 48 hours. That's what they're predicting, the mRNA. After that, it gradually moves out of it's degraded, and it's not available for the ribosome to make this specific spike protein. And because of the mRNA platform, it's a very specific antibody that is going to be made, protein that is going to be made, and that creates a specific antibody that's made because of the spike protein. Coronavirus, I think it has about 28 different proteins. So if you have a primary infection, if you're actually getting a virus in your body, a coronavirus, when the body is going through that immune system, it is going to make 28 different antibodies in your body, correct? Because of that, some of the exposed proteins would not be even beneficial for the immune response. So you are using up so much energy without focus to make 30 odd antibodies, whereas now with the vaccine, you are making a very specific one protein specific antibody. So it's a more controlled reaction, and the downstream is also more controlled. It's not an acute reaction. But if you are getting exposed to the primary virus, you are making 28 to 30 different proteins and creating 30 odd antibodies. That's where that cascade of overreaction of the immune system takes place. That's when people end up in ventilators. So that's why the folks who got the vaccine in these phase 3 studies and the people who are getting the vaccine now, even if you get exposed, and even that 6% chance makes you get infected by a virus, you already have a more controlled reaction in your body, reducing a chance of acute infection and also hospitalization. So it's you get certain discomforts, you get fever, sometimes like itchiness, or the area becomes a little warm to the touch. So these are all aspects of an immune reaction that is taking place, but it does not make it an acute infection. And sometimes people discuss, oh, how is this going to test my qPCR COVID test? Because you are making an antibody and the mRNA is not there forever, it's get broken down. The PCR test does not get affected. Now, if your vaccine is doing its job and it's actually making antibodies, if somebody is taking an antibody test as the COVID test, then yes, you will become positive because you have generated antibodies, in this case, which is a good thing. So Paul, I cannot do a presentation without putting an image from a microscope. This is to show you some of the viruses inside your body. This is a transmission microscope. Forgive me, Paul, if I'm butchering the introduction for the image. And these are the little round structures are the viruses attached to cell membranes here. And I'm not familiar with this structure, but this is to show you how a virus looks like within the host after infection. Okay. So we have 9 approved vaccines globally right now, and they are based on, some of them are based on the mRNA that we just discussed about. Then we have some inactivated vaccines. Remember what we discussed about traditional groups of vaccines. And these are also vector vaccines where we package that specific message for a spike protein using a skeleton of a virus that is no longer replicating or no longer creating an acute infection. If you want to visualize it, if you take a car, and take out the steering wheel, take out the seats, and pack them with balloons, okay? From inside, outside, it looks like a car, but inside, it's not a car anymore. So people take a skeleton of the adenovirus and then package it with a message that is specific for the spike protein presentation of COVID. And then you infect that adenovirus, and the virus will get into your body. And because of the outside has that viral particulate, it is at an advantage where it compared to mRNA. So not only it has a more controlled reaction for the spike protein that we are packaging, the message, the adenovirus itself will present. So the other immune cascades are generated by the adenovirus as well. So your immune system is at a more of an alert condition. So that's an advantage of using a vector virus versus the mRNA. But one of the mRNA advantages is because it's an mRNA sequence that you're manipulating, I'm going to use the word easily, which is much more complicated, right? But you can, based on different variants of the virus that are generated, we can change the sequence. So if there's a mutation on the spike protein, we can change the mRNA sequence. That's why these companies like Moderna and BioNTech, they're already working on different strains or variants of that, the viruses that are available globally. If you have heard about the British version, the South American version, now there's a Brazilian version of it, so they can manipulate these things and quickly have another booster available with change sequence that will be more effective against a new strain. Okay, so these are the mRNAs that we discussed about. Okay, so what is the need for COVID? Okay, you as a future scientist or future administrator, you need to read about both sides, right? So, but I'm of course more biased, and I'm going to present you the data. Okay, so these numbers probably have gone up. This is probably a week ago. Okay, we have about 104 million people, and out of that, about 2 million deaths. Be significant. That's about 76% people who recovered as well. Okay, but look at the economic, the hit people are getting. You're getting about trillions of dollars to fight COVID, the pandemic, and to get a patient through the hospital system, it's 500 times more to fight COVID, putting people on the ventilator, giving them medicines, taking care of them versus prevention. In the United States, we have about 27 million cases and 460,000 folks dead. And the healthcare, it's about $30,000 to $50,000 per hospitalization. So I know all of you are aware of the mathematics of it. It's a significant hit. So we have these vaccines, okay, and it is preventing, remember I said 100% of the people, whether you got infected or not, those folks did not have to go into a hospital. And now we have about 22 million total doses in California, and it creates about 18% of the population is fully vaccinated as of 2 days ago. So things are looking good, looking upward. And this is to show you where US and how globally the patterns of it. Some of the countries are doing very well, and USA is lagging behind, but the trend is well. It's increasing as a total population, okay? And this is to give you an idea, rest of the global. So are we on a hard stop for 4 o'clock? Okay. All
yeah.
right. Okay.
I just want to say, it seems like it looks like we're on track, right? With when
Okay.
it's coming down to
Yeah.
all the vaccines.
So this is what the predictions are with 3 vaccines. We can account for a complete vaccination of the entire population. Pfizer is already working on dosages for age 12 to 15. So now it's open for 16 and above. So pretty soon the entire population should be vaccinated. I'll stop there. How's that? Okay.
No, that was wonderful, really great information that's super valuable and powerful. And so sorry to cut you off. But I really appreciate the whole presentation, and I'm sure that the students and the teachers do as well. So one more time, thank you, Dr. Webster, and thank you, Manji. Again, very detailed presentation and educating us all on the immune system and obviously all the vaccines and everything that's going on. Again, very powerful, very clarifying for all of us. Thank you, students and teachers, for your questions and comments. And just so you know, there's going to be another event coming up on this COVID series, and that's going to be on antibodies and antigens. So we will continue to elaborate on this discussion. That will be held on Thursday, April 22nd from 3 to 4 p.m. and we hope to see you there. Students, if you have not signed in, please make sure you sign in using the link that's on the chat box. And teachers, please make sure you now sign out. And that is it. With that being said, have a great rest of your Thursday. Thanks again, everyone.
All right, cheers.